Today is day 2 of our discussion of metabolism, and we're going to start with lipid metabolism. The most abundant lipid in the body are triglycerides. And the body can use triglycerides to produce energy. So lipids, like triglycerides, will go through the process of lipolysis to produce ATP. They will either get broken down into pyruvate, or they will get broken down directly into acetyl-CoA. If they're broken down into pyruvate, pyruvate will be broken down into acetyl-CoA, just like we saw in glycolysis where a carbon is removed, and then acetyl-CoA will move into the citric acid cycle. Then all those hydrogen from the citric acid cycle will be taken to the electron transport system to create the conditions to make ATP. So let's take a look at what a triglyceride is. A triglyceride has a glycerol head, and it has 3 fatty acid tails. Now these fatty acid tails are chains of carbons. When the triglyceride gets broken down, it'll be broken down into the glycerol head and the 3 individual fatty acid tails or chains. So let's take a look at that molecule then that's been broken down. We have the glycerol head and the 3 fatty acid chains. And so from one fatty acid tail, if it contained 18 carbons, so we see that the fatty acid tails, these fatty acid chains, actually hold a lot more carbons than what glucose did. Glucose only had 6 carbons. This fatty acid tail would on average have about 18 carbons. So from one 18 carbon fatty acid, that would yield 9 acetyl-CoA. And from 9 acetyl-CoA, that will yield around 90 to 96 ATP from one fatty acid tail. Now that is 1.3 times more than glucose gave us. So if you remember, glucose was only 6 carbons. So each of those carbons on glucose are going to have hydrogen attached to them, which can be removed and carried off to the electron transport system, where they can be used to create those conditions to make ATP. The fatty acids have 18 carbons in those tails, so they just have a lot more hydrogen that will help to yield more ATP. So in order to yield the same amount of ATP, you would need 3 glucose molecules for every 1 fatty acid to yield the same amount of ATP. Okay, let's talk a little bit about lipid anabolism. Okay, so lipogenesis, lipid formation of, right? This is the formation of lipids or the formation of triglycerides. Triglycerides are stored in adipose tissue. So when does your body form triglycerides? Well, we know that it is stimulated by a diet high in carbohydrates, it's also stimulated by the hormone insulin. And insulin will be released whenever you have a diet high in carbohydrates. Some lipids can be made in the body, just like we had with glucose. We saw glucose could be produced through the process of gluconeogenesis. And so some lipids can be produced in the body, but some lipids can't be produced in the body. For example, the linoleic and linolenic acids. These lipids come from plants, and so they are not produced in your body. They can't be produced in the body. So any of the lipids that you have to get from your diet, those are called essential fatty acids. The ones that your body produces are called non-essential fatty acids. So non-essential fatty acids are fatty acids your body can produce. Lipids are stored as little compact molecules. So they're very hard to get at, so the body doesn't really like to use those first. They're processed in the mitochondria where oxygen is limited, and the lipids need to have oxygen in order to create those ATP. So this isn't going to be the first choice for your body to use. Your body's first choice for fuel is glucose. But glucose metabolism will shift to lipid metabolism when glucose supply is low. It also will shift to lipid metabolism when energy demands are low as well. All cells need lipids to maintain their plasma membranes, so lipids are very important to the cells. So fatty acids are going to be delivered to the cells in the body. In the blood, there are free fatty acids just floating around in the blood, but they're only a small percentage. This small percentage of fatty acids might bind to albumin, but it's a very small percentage. Most lipids circulate in the bloodstream as lipoproteins. Especially cholesterol. So let's talk about what lipoproteins are. Lipoproteins are a mixture of cholesterol and glycerides, monoglycerides, diglycerides, triglycerides, and they are coated by phospholipids in proteins. And there's different classifications of lipoproteins, so I just want to go over what those are now. The first one is a chylomicron. These you've heard of before. They're the largest ones that there are. They make up 95% triglyceride. These chylomicrons are produced when you absorb triglycerides from your diet into the cells of the intestinal wall, and then the triglycerides are made into chylomicrons. And from there, the chylomicrons move into the lymphatic system. The second type of lipoprotein is called a very low density lipoprotein, or VLDLs. VLDLs are, again, mostly triglycerides. And They transport the triglycerides to skeletal muscle and adipose cells. Where they'll get stored and then they can get used as energy as needed. The next type of lipoprotein is a low-density lipoprotein, or LDLs, and you've probably heard of these as the bad cholesterol. They are mostly cholesterol. They deliver cholesterol to the peripheral tissues. They can end up in your arteries as arterial plaque. The last type of lipoprotein are the HDLs, and a lot of times that's high density lipoproteins, and they are called the good cholesterol. They are mostly cholesterol. And phospholipids. They transport excess cholesterol from the tissues back to the liver. And then the liver can either store them or excrete them. So let's use this picture here to talk about these different lipoproteins. So here we have the chylomicrons. These are the ones that are produced when you are absorbing the fats from your diet. So the triglycerides from your diet get absorbed into your cells of your intestine and then are packaged up as these chylomicrons. The chylomicrons get absorbed into the lymphatic system, and then the lymphatic system drains into those subclavian veins. So now the chylomicrons are in the blood. So the chylomicrons travel through the blood until they get to the liver, and then they are dropped off in the liver, and the liver gets to decide what to do with them. So the chylomicrons are broken apart, and the triglycerides from the chylomicrons are used to produce VLDLs, so very low density lipoproteins, and those VLDLs will carry those triglycerides off to the skeletal muscle and to the adipose tissue. And some of the triglycerides from the chylomicrons will be used to produce these LDLs, or low density lipoproteins. So mostly what LDLs are made up of is cholesterol. Cholesterol is part lipid, part protein. The LDLs carry this cholesterol to the peripheral tissues. So here we see the LDLs traveling to the peripheral tissues and then moving inside the cells. And inside the cells, the LDLs are broken down. The cholesterol is stripped off, and then the cells can use the cholesterol. Any excess cholesterol then will go back into the blood system or back into the bloodstream, will be absorbed by the HDLs. The HDLs then will return that cholesterol to the liver, and that excess cholesterol is extracted, and then it's packaged into new LDLs. The HDL then leaves the liver, goes back out into the circulation, and picks up the excess cholesterol again and delivers it back to the liver again. So the HDLs are really healthy. They're taking any excess cholesterol and taking it back to the liver to be repackaged. That's so that the cholesterol won't pack into your arteries and cause atherosclerosis. Cholesterol does have many vital functions in your cells. First of all, it is a precursor for hormones. Especially those steroid hormones like estrogen and testosterone. It's a precursor for vitamin D. We need vitamin D to absorb calcium. It helps to waterproof the epidermis. It is found in cell membranes. It's found in bile. So cholesterol has many, many functions in the body. The liver makes about 20% of your cholesterol. So even if a person really reduces their diet and tries to take as little cholesterol in as possible, their liver might still be producing the cholesterol. So the dietary guidelines say that you should eat as little cholesterol as possible, no more than 300 milligrams per day. Some people have a genetic predisposition for cholesterol, that if they reduce their cholesterol intake, then the liver will just make more. So someone that has this genetic predisposition might not have much luck reducing their cholesterol levels in their body just by reducing the amount of cholesterol that they have in their diet. Cholesterol levels in the body varies with age. If we look at a fasting cholesterol level, a person at age 19, anything over 185 milligrams per deciliter would be high. But for the same person at age 70, anything over 250 milligrams per deciliter would be high. For females, it's a little bit different too. Anything over 190 at age 19 is high, and anything over 275 at age 70 would be high. Let's go over some blood testing. So when your blood is drawn, usually you're fasting to look at lipids, and a lipid panel, is a test that would be performed. And this lipid panel would look at your total cholesterol levels. It would look at your HDL. It would look at your LDLs, and it would look at your triglycerides. So the desirable amount, we'll just go over the desirable amounts right here. The total cholesterol desirable amount would be less than 200 milligrams per deciliter. A normal HDL level would be anything greater than 60 milligrams per deciliter. Normal LDL levels would be anything less than 100 milligrams per deciliter. and normal triglycerides would be anything less than 150 milligrams per deciliter. The LDLs are determined through an equation. So the LDLs are determined by the cholesterol minus the high-density lipoproteins, or HDLs, minus the triglycerides divided by 5. So let's look at the LDLs of a person that's like right on the border. So if we have LDLs and cholesterol of 200, and we subtract the HDLs, which is 60, and we subtract the triglycerides, which we'll say is 150, and we divide that by 5, we get 200 minus 60, which is 140, minus 30, which is 110. So in this case, a person would have 110 milligrams per deciliter of LDLs. And according then to this chart here, the LDLs would be slightly elevated. When LDLs are elevated, then dietary changes and even drug therapy would be recommended. A lot of times they'll recommend these drug therapies and dietary changes anything over 100, but definitely over 130 milligrams per deciliter. So now let's start talking about protein metabolism. As we know, proteins are made of amino acids, chains of amino acids. Amino acids are linked together by peptide bonds. Right? And so these peptide bonds would have to be broken in order to liberate one of the amino acids in order to break that down to use it for energy. There are only 20 different amino acids in your body. And from those 20 different amino acids, we can rearrange them. We can have different lengths of them by adding more amino acids. And so with just the 20 different amino acids in the body can make 100,000 to 140,000 different proteins. So to get ATP from one amino acid, the amino acid has to be converted to a different substance. So an amino acid would be converted to glucose or a fatty acid or a ketone body. And then these things can enter into the citric acid cycle. Right, so there's 3 different processes to do this conversion. We're going to talk about each one of these. There's a process called transamination. There's a process called deamination. And then there's a process called the Urea Cycle. We'll start by talking about transamination. Transamination is a process that the cells of the liver and skeletal muscle, cells of the heart, the lungs, the kidney, and the brain all use to convert amino acids to glucose. In transamination, an amino group is removed from an amino acid and then it's transferred to a keto acid. then we end up with a new keto acid and a new amino acid. The new keto acid enters the cytosol, and the new amino acid enters the citric acid cycle. So let's take a look at what I'm talking about. So we have an amino acid, and let's just say that amino acid is glutamic acid. Okay, so the body can't use that. Glutamic acid can't enter into the citric acid cycle, so it has to go through this process called transamination. And an amino group is taken off of this amino acid, and it's transferred to a keto acid. So an amino group is transferred. So now we have a keto acid 1 will bond to this amino group and it'll become a different keto acid. And the amino acid loses that amino group and it becomes another amino acid. And so the amino acid that it would become is tyrosine. Now tyrosine is an amino acid that can enter into the citric acid cycle. So that goes through the citric acid cycle, and then that produces ATP. The other keto acid then, keto acid 2, is just going to enter into the cytosol from the mitochondria. The second process is called deamination. During deamination, an amino group is removed again, but in this process, it's not involving the keto acids. Instead, by removing an amino group, an organic acid is formed. But at the same time that the organic acid is formed, ammonia is also formed, and the ammonium ion is toxic. It's toxic to the cells. So then the liver enzymes have to convert the ammonium ion into urea, which is not toxic. So let's look at this. So say we have an amino acid, and we'll use glutamic acid again. And this amino acid goes through the process of deamination. And after it deaminates, what that means is it's losing, so it loses an amino group. And then it becomes an organic acid. So this organic acid can be used by the cell, right? But it also generates the ammonium ion. And the ammonium ion is toxic. So the process of deamination does create a useful acid, but it also creates a toxin that the body has to get rid of. So then the liver enzymes have to get rid of that. They're going to do that in that next cycle, which is called the Urea Cycle. So there is a genetic disorder that I want to talk to you about. It's called PKU, or in other words, it's called phenyl ketone urea, PKU. Normally, the amino acid called phenylalanine is converted to very useful hormones called norepinephrine, epinephrine, dopamine, or melatonin. And if you remember, these hormones are amino acid derived hormones. So they are basically amino acids. Phenyl ketone urea, or PKU, is a rare inherited disorder that causes phenylalanine to build up in the body. So PKU is caused by a defect in the gene that helps to create the enzyme that breaks down phenylalanine. So phenylalanine starts to build up because there's no enzyme that will break it down. And too much phenylalanine in the body causes problems with the brain and other organs. Damage from a buildup of phenylalanine can begin within the first month of life after a baby is born, and if undetected or untreated, it can result in severe mental retardation, hyperactivity, and seizures. So let's now take a look at how we get rid of this ammonium ion. When the ammonium ion is produced from the deamination of amino acids, we have to get rid of that ammonium ion because it's toxic. And so ammonium ion will enter into what we call the urea cycle. And in the urea cycle, it combines with carbon dioxide. And together then they produce a waste product that's called urea. Urea is not toxic, whereas ammonium ions were very toxic. So now the kidneys can excrete urea. So those are the three processes that occur with protein catabolism. The body likes to break down proteins only when the glucose supply is low. So basically, proteins are not a great source of energy. First, glucose levels will be broken down to produce ATP, and then lipids will be broken down to produce ATP, and proteins will only be broken down if the glucose supply is low and if the lipids are inadequate. So the liver is going to be breaking down these proteins, and they break down internal proteins within the liver. So breaking down proteins to produce ATP is actually very impractical. There are 3 reasons why it's impractical to use proteins for energy. So first of all, proteins are more difficult to break apart. Second of all, the byproduct ammonium ion is toxic. So we wouldn't want to break down proteins for that reason. And the third reason is that breaking down proteins threatens homeostasis. You're basically breaking apart the cells of your body in order to produce ATP. So that's going to cause some type of organ disorder, organ failure. So it's not a great idea to try to use protein to get your energy. Like I said before, there are 20 amino acids in your body, and 10 of those amino acids come from your diet. And so those we call essential amino acids. Think of it this way, it's essential to eat these and get these, otherwise you're not going to have them. Your body can't make them, so it's essential that you eat them. Of these 10, 8 of these your body cannot make. And 2 of these your body doesn't make in a high enough quantity for a growing child. Then 10 of these are non-essential amino acids. And these are amino acids that your body can make through anabolism. And when we are producing new proteins, that process is called amination. So your body has to decide when it's going to break down these nutrients and produce energy, or when it's going to store these nutrients for a rainy day. There are 2 processes in your body, 2 states in your body. One is called the absorptive state, And in this state, that's when your energy is being stored. So for glucose, for example, instead of breaking down glucose, this is when you're going to be storing glucose as glycogen. Another state in your body that you have is called the post-absorptive state. The post-absorptive state is when your body is going to release energy. So this is when your liver will take glycogen and break it down into glucose, so then glucose can become available to make ATP. There are several organs that are involved in trying to figure out whether you should be storing your nutrients or whether you should be releasing energy. So the first one is the liver, and the liver is really the focal point. The liver enzymes will break down or make carbohydrates, lipids, and amino acids. There's an extensive blood supply going to the liver, and the liver will determine what quantity of carbohydrates, lipids, and amino acids are in the blood, and then they can monitor and adjust those nutrients accordingly. So for example, if the blood supply coming to the liver is low in carbohydrates, low in glucose, then the liver has the ability to break down glycogen into glucose and release that into the blood. It also has the ability to go through gluconeogenesis, where it is creating new glucose molecules. Right? So the liver has the ability to do those things. The liver also has a significant energy reserve in glycogen. So glycogen is stored in the liver. And glycogen, like we said, can be broken down into glucose that will be released into the blood to resupply that if the blood is low in glucose. On the other hand, if the blood is high in glucose, as it's passing by the liver, then the liver can take that glucose and store it as glycogen. Another tissue is adipose tissue. Adipose tissue stores triglycerides. Adipose tissue is found in areolar tissue that's right under the skin. It's found in the mesenteries surrounding the intestines. It's found in the bone marrow, and it's found in epicardium. So it's in plenty of places. When the body's at rest and it has more time, it'll pull those triglycerides out of the adipose and break down those triglycerides to produce ATP. That's only going to happen when energy demands are low and the body's at rest, or if glucose levels are low. Another organ that is involved in this process are skeletal muscles. Skeletal muscles are about half of a person's weight. So they are contributing to a lot of a person. The skeletal muscles have large glycogen reserves. When those skeletal muscles need glucose to produce ATP, so the skeletal muscles can contract and get you out of a difficult situation, then that glycogen will be broken down into glucose. And if necessary, if the glycogen levels run out, so you don't have glucose, then the skeletal muscles will sort of eat themselves, and they will break down the contractile proteins. So you remember what the contractile proteins are. That's like the actin and myosin. So if the skeletal muscles have to, they will break down the contractile proteins, which is not good, but will happen in situations where a person is starving themselves, or when a person is in that last stage of the general adaptation syndrome or the stress syndrome. Another tissue involved is nervous tissue. Nervous tissue has a high demand for glucose. So nervous tissue it can't really store carbohydrates, lipids, or proteins. It doesn't store those, so it can't maintain a reserve. So if a person has low glucose levels, then they will become, if it's too low, they will become unconscious, because the nervous tissue just doesn't really have any reserves to pull out glucose from to create ATP. And it really can only use glucose. So it needs to have those glucose levels within a certain range. And then there are other peripheral tissues that are involved. Not as much. They don't have large reserves. But if they need to, they can metabolize glucose and fatty acids. So they can break down glucose and fatty acids if necessary to produce ATP. Let's talk about a 24-hour period in your day. Within a 24-hour period, you have an absorptive state and you have a post-absorptive state. The absorptive state, like the name implies, you're absorbing nutrients. This state follows a meal, and your cells absorb the nutrients. The post-absorptive state comes hours after a meal, hours later. And this is where the cells are going to focus on maintaining the blood glucose levels to meet the needs of the nervous tissue. So in a way, we could say that the absorptive state is the harvest time. So we are harvesting the crops and we're storing them in the barns. And then the post-absorptive state, this is going to be between harvests. So this is where we're pulling out our storage and we are using what we had stored during the harvest stage in order to maintain our energy levels. Let's go a little bit further into each one of these. So in the absorptive state, this is going to last about 4 hours. If you have 3 meals a day, that means that you are in this absorptive state 12 hours a day, right? In this state, insulin is going to be the primary hormone, because what insulin does is it binds to your cells and it allows your cells to absorb the nutrients. It allows your cells to absorb the glucose and the lipids and the amino acids. Growth hormone is also involved. Growth hormone stimulates the amino acid uptake by your cells as well as protein synthesis. So it's allowing your cells to bring in those amino acids and then to make proteins with them. So in the absorptive state, fatty acids and glycerol will be stored as triglycerides. And then amino acids will be stored as protein. And glucose is stored as glycogen. And acetyl-CoA can also be stored. It will be stored as a fatty acid. Right, let's look at the post-absorptive state. In the post-absorptive state, nutrients are not absorbed. In this state, if you had those 3 meals a day, then anything in between those 3 meals a day will be in post-absorptive. So the post-absorptive state would take about 12 hours out of your day. If you skip meals, it will extend this. So if you only have 2 meals a day and each meal is in the absorptive state, you're in the absorptive state for 4 hours for each meal. So if you only have 2 meals a day, you're in the absorptive state for 8 hours, which means you'd be post-absorptive for 16 hours in a 24-hour period. During the post-absorptive state, you mobilize energy. And you do this to maintain glucose. There's hormones that are involved like glucagon and epinephrine and glucocorticoids. And growth hormone. Glucagon stimulates the conversion of glycogen to glucose. So it releases more glucose into the blood, which will increase blood glucose. Epinephrine also stimulates the conversion of glycogen to glucose to increase blood sugar. Glucocorticoids stimulate the process of gluconeogenesis. Gluconeogenesis is the process of the liver making new glucose. And growth hormone also stimulates gluconeogenesis. And it also inhibits insulin. So if it's inhibiting insulin, that means that glucose can't get into your cells, and if glucose can't get into your cells, then glucose will remain in the blood and you'll have an increased blood glucose level. Again, the liver cells want to break down glucose because glucose is easy and they're going to be able to break it down much faster. But if glucose isn't available, then the liver cells are going to break down lipids and amino acids for energy. So the liver will break down lipids and amino acids if glucose isn't plentiful, or if the body demands increase and there's not enough glucose for the body demands, or if the body is at rest. When lipids are broken down, they form ketone bodies. Ketone bodies are kind of an alternate tool for glucose, if glucose is in short supply. So basically, ketone bodies are formed when there's not enough sugar or glucose to supply the body's fuel needs. This will occur overnight, or it also occurs during dieting or fasting. These ketone bodies can then get converted to acetyl-CoA. And then acetyl-CoA can enter into the citric acid cycle to produce ATP. If a person is healthy, then their liver produces ketone bodies in a low concentration. And so then this would not be detectable in a urinalysis. However, when a person is fasting, they have an increased production of ketone bodies. And so now we have more ketone bodies than we need, and this leads to what we call ketosis, which is a high concentration of ketones. Ketonemia is a term that we used for high ketones in the blood. So whenever you see EMIA, emia, that means in the blood. So usually ketonemia is controlled by buffers so that the levels of ketones in the blood stays low. Usually a normal ketone concentration would be under 200 milligrams per deciliter. If the pH of the body drops below 7.05, then more ketone bodies are going to be produced, and you'll have ketonemia. And ketonemia, if it's severe enough, can disrupt tissue activities. It can cause coma. It can cause arrhythmias of the heart. It can even cause death. The nervous system will metabolize ketone bodies if it has to. After like long periods of starvation. So this is the order of what will be used. First of all, glycogen reserves will be broken down. So glycogen reserves are used first. They're broken down into glucose, and then glucose can be used to produce ATP. Second, gluconeogenesis will occur. This is where the liver makes new glucose. And then third, if the glucose levels still are not where they should be, then the lipids get broken down, and the lipids produce ketone bodies. And amino acids are catabolized or broken down, and they produce ammonium. Ammonium, we already know, is toxic to the body, and ketone bodies. Another thing that we know about ketone bodies is in a diabetic who has uncontrolled diabetes. So what I mean by that is that they really have low insulin or no insulin levels. When you don't have enough insulin, that means that your cells can't get to the glucose. So if we look at your cells, I'll just draw a cell here, and there's these glucose channels, right? And glucose is in the extracellular fluid, and glucose wants to come into the cell, but it has to, first of all, you have to have insulin binding to the receptor. And if insulin binds to the receptor, then the gate opens, and then glucose can come into the cell. But if you don't have insulin, then that gate is going to remain closed, and glucose will not be able to get into the cell. It can't get in, right? So the cell still needs energy, so what the cell's going to do then is it's going to break down lipids. That's what it wants to do first before it breaks down amino acids. So if lipids are going to be broken down, they're going to produce ketone bodies. We're going to learn later that ketone bodies then will be converted to keto acids. And if there's too many keto acids, then we end up with a ketoacidosis. Thus the term diabetic ketoacidosis, which is a dangerous condition that a person has if they have uncontrolled diabetes. All right, let's talk a little bit about nutrition. So a person should have a balanced diet, meaning they should be eating the proper nutrients. So a balanced diet just means that you are receiving all the ingredients for homeostasis. Malnutrition, then, on the other hand, means that you are getting an inadequate or you're getting an excess of nutrient absorption. There are 5 different food groups, and I know you've heard these before. There's grains and veggies and fruits and dairy and proteins. A complete protein is a protein that provides all the essential amino acids in sufficient quantities. Things like beef and fish and poultry, eggs and milk, they're all considered complete proteins because they can provide you with all the essential amino acids in sufficient enough quantities. An incomplete protein is a protein that is deficient in one or more amino acids. So sometimes vegetarians, they have a diet that's restricted to grains and veggies and fruit. They might have a diet with incomplete proteins. Nitrogen is an atom that we look at in our balanced diet. And so the amount of nitrogen that you eat or bring in through what you eat or drink should be the same amount of nitrogen that you excrete. And if they're the same amount, that means that you are in nitrogen balance. But if you are eating and drinking more than what you're excreting, then you have a positive nitrogen balance. So in other words, if you bring in more than what you excrete, you have a positive nitrogen balance. On the other hand, a negative nitrogen balance means that you are excreting or getting rid of more than what you are bringing in. I mention nitrogen because nitrogen is important. Nitrogen is found in your amino acids, it's found in your DNA and your RNA, it's found in creatine. Creatine is an energy storage for muscles, and it's in porphyrins. Porphyrins are those little bullets that the cytotoxic T cell and the natural killer cell put out, those little bullets. So nitrogen it's important. On the other hand, nitrogen should really be in balance. You want to make sure that you're eating and drinking as much as you're excreting. You don't want to be in a positive nitrogen balance or a negative nitrogen balance. You really want that nitrogen to be balanced out. The only time you would want a positive nitrogen balance would be if you had a person that was an athlete who needed more nitrogen, or if in a pregnant woman who needs more nitrogen, or even in a growing child. These are all examples of when you would want to have a positive nitrogen balance where you're bringing in more than you are excreting. Let's discuss some of the minerals you should have in your diet. Minerals are inorganic ions. So we're talking about like sodium and chloride and potassium. Sodium and chloride, they determine the osmotic pressure or the osmotic concentration in the extracellular fluid. So we know that sodium is more abundant outside the cell, and so is chloride. And so sodium and chloride are going to draw water towards them. And so how much sodium and chloride you have in that extracellular fluid is going to determine the osmotic concentration. Potassium, on the other hand, this determines the osmotic concentration of the intracellular fluid. So we know that potassium is higher inside the cell, and so how much potassium you have inside the cell determines the osmotic concentration inside the cell. So first of all, I guess what I'm saying here is that one of the functions of minerals is to determine osmotic concentrations. The 5 major minerals in the body are calcium, phosphorus, potassium, sodium, and magnesium. Okay, so I just talked about potassium and sodium. So now I'm just going to discuss the other functions of minerals. And so there's quite a few functions of these 5 major minerals. It maintains the membrane potential. They maintain the membrane potential. So if you remember what the resting membrane potential is, it's that negative 70 millivolts inside the cell, right? Negative 70 millivolts. So sodium and potassium really do that. These minerals are necessary to construct the skeleton. So calcium and magnesium are going to do that. They're necessary for muscle contractions. Calcium is definitely necessary to contract your muscles. They are necessary to generate ATP. They are necessary to release neurotransmitters. Remember that calcium is necessary to release neurotransmitters from the synaptic terminal. They are necessary to produce hormones. They're necessary for blood clotting. They're necessary for the buffer systems of our body. We'll see how phosphorus is used in some of the other ones. It's necessary for fluid absorption, right? When sodium gets absorbed, water follows. And also for waste removal. which are all things that we study when we study the systems of the body. And the third thing that I want to mention about these minerals is that they're necessary for enzymatic reactions. So, for example, ATPase. ATPase was that enzyme in the electron transport system that is the gate that hydrogen moves through and activates the phosphorylation of ADP plus phosphate into ATP. And for this enzymatic reaction, we need both calcium and magnesium. Those are both necessary for that enzymatic reaction. There's another enzymatic reaction that requires zinc. Zinc is another mineral. In that process where carbonic acid is broken down into water and carbon dioxide, that requires an enzyme called carbonic anhydrase. Carbonic anhydrase needs zinc. Copper is another mineral that's needed for the electron transport system. It is actually needed for that last cytochrome. So remember, in the membrane we had those proteins, right? So copper is needed for that final cytochrome, which accepts the electron and then releases it. Iron is another mineral. We need iron to make red blood cells. They make the heme group in the red blood cells. And so iron deficiency means that there's not going to be enough red blood cells being made. And iron deficiency is more common in women. Because women have a lower reserve than men do. And so a deficiency is more likely to develop in a female than it is in a man. I've mentioned vitamins before, but I'm going to mention them again here. Vitamins really function as coenzymes, so they are important. They're necessary for enzymes to function properly, basically is what that's saying. There's fat-soluble vitamins, and then there's water-soluble vitamins. The fat-soluble vitamins are vitamins A, D, E, and K. They're fat-soluble because they dissolve in fats, or they dissolve in lipids. And they are mostly absorbed in the GI tract. Okay, but they can also be absorbed from the sun. We know vitamin D is absorbed from the sun. A couple of specific things that you should know is that vitamin A is needed for visual pigments. So they're necessary for those receptors in your eye. Vitamin D is necessary to vitamin D gets converted to calcitriol, and calcitriol then allows us to absorb calcium. So basically, vitamin D aids in calcium absorption from the gut. And then vitamin K is necessary for blood clotting. So those are some pretty important vitamins. Then we have the water-soluble vitamins, and these are also coenzymes. We have niacin. Niacin is used to make NAD. NAD is that carrier that will carry hydrogen. B2 is a water-soluble vitamin. Another name for that is riboflavin. Riboflavin helps to make FAD, another hydrogen carrier. And then we have pantothenic acid, which is B5, and that helps to make coenzyme A. Water-soluble vitamins are rapidly exchanged between the fluid compartments and the blood, so they just go very quickly between fluid compartments. And the other thing is, probably more importantly, is that the water-soluble vitamins are excreted very readily by the kidney. Because they're so readily excreted by the kidney, that means that hypervitaminosis is rare. Hypervitaminosis means that you have too many of these vitamins in your blood. And because they're excreted readily by the kidneys, that's not really going to happen. There are other B vitamins that are water soluble, so I want you to know those. I want you to at least know the names of these vitamins. So B1, another name for vitamin B1 is thiamine. Another name for vitamin B2 is riboflavin. B3 is niacin. B5 is pantothenic acid. B6 is pyridoxine. B9 is folic acid. B12 is cobalamin. B7 is biotin. And then another water-soluble vitamin is vitamin C, otherwise known as ascorbic acid. Right, so I did give you some of the functions of these. We'll just go back over that quickly again. B2 or riboflavin is necessary to make FAD. Niacin is necessary to make NAD. Pantothenic acid is necessary to make coenzyme A. Pyridoxine is necessary to make amino acids and lipids. It's necessary for amino acid and lipid metabolism, and so is folic acid. They're both necessary for the breakdown and the production of amino acids and lipids. Cobalamin is necessary for nucleic acid metabolism. And then biotin and ascorbic acid and thiamine, they're all necessary coenzymes in enzymatic reactions in the body. And again, if you have if you're taking in more than what you need, that's called hypervitaminosis. And if you're not getting enough, then that is called hypovitaminosis. So your metabolic rate is what I want to talk about next. And your metabolic rate is your daily energy usage. So if your intake of nutrients exceeds your demands for those nutrients, then you're going to store that energy in adipose tissue. But if your body's demand, because you have increased activity, if that exceeds your daily intake, then you will use your reserves and you'll lose weight. A person who is sedentary has minimal energy demands. So they most likely will be taking in more than their body demands, and they will store these nutrients. But in people that are active and doing things like activities like swimming, for example, then they will have significant energy demands. And that means that their demand will be much greater than their intake. Calorimetry measures the energy that's released when bonds are broken. So we're measuring energy through calorimetry. A calorie with a small c is the amount of energy required to raise 1 gram of water 1 degree Celsius. Since humans are weighed in kilograms, though, and not in grams, we would convert the calorie with a little c to a calorie with a capital C. Which means kilocalories. It's just a conversion. Food contains calories. We can determine how many calories a particular food has by putting it in a chamber called a calorimeter that's filled with oxygen and water, and then that food is burned. And then the number of calories released is recorded. And that energy potential of the food then is expressed in kilocalories per gram. And by doing this, we find out that lipids, like we would expect, has 9.46 kilocalories per gram. And so they are definitely the most productive. If we compare lipids to carbohydrates, carbs only yield 4.18 kilocalories per gram, and proteins only yield 4.32 kilocalories per gram. The BMR, or basal metabolic rate, is the rate of energy expenditure while at rest. So how much energy are you expending at rest? For example, your energy needed to breathe and to keep warm. On average, the BMR should be 1 kilocalorie of energy every hour for every 1 kilogram of body weight. So a man that has an average weight of 160 pounds would weigh 72.57 kilograms. So that man should be burning 72.57 kilocalories of energy each hour at rest. The BMR is affected by different things. It's affected by exercise. If a person is exercising, their basal metabolic rate is going to be higher. They're going to be burning more than 72.57 kilograms each hour. It's also affected by age. The older a person is, the less they're going to be burning each hour. Sex is also a factor. Males are going to burn energy faster than females. Hormones are also involved. For example, thyroid hormones. If a person is producing more thyroid hormones, they're going to be burning energy faster. Endocrine activity. There's some hormones that will increase energy usage. And then also climate. People that live in warmer climates are going to be burning energy faster than those in colder climates. Another thing is that the BMR is what your basal metabolic rate would be at rest, right? So if you are taking that measurement while a person is sprinting, then that's going to be a much different measurement than if you take that measurement when a person is sleeping. For that reason, when we're measuring the BMR and studying the BMR, we want to standardize the testing conditions. So a person, when you're measuring the BMR, should be awake and alert, but resting. For a person that has a BMR of 70, which I showed you with a 160-pound man, then they will be, that's per hour, 70 kilocalories per hour. That means on a daily basis, they would be burning 680 kilocalories per day. Their physical condition, their body weight, their genetics are all things that can influence the BMR. If a person has a low BMR, they may develop obesity. Obesity is when a person has 20% more body weight than normal. And so we need to look at obesity as being an important health factor, because obesity can cause things like heart disease, it can cause cancer, and it can cause diabetes. The last thing that I want to talk to you about in this chapter is called Thermo Regulation. So energy that's produced from breaking bonds is captured by your cells as ATP, or it's released as heat. So in other words, when glucose is broken down, all I've been talking about is that glucose will produce ATP. But actually, glucose getting broken down produces both heat and ATP. Heat helps to keep the body temperature normal, so it helps to maintain the body temperature. The enzymes are going to work when the body temperature is normal, so it's important to have normal body temperature. Anything below 97 degrees Fahrenheit or above 104 degrees Fahrenheit will cause disorientation. Person will be dizzy, confused, disoriented. Anything above 108 degrees Fahrenheit will cause convulsions and permanent cell damage. So basically, this heat is produced as a byproduct of metabolism. And then the heat is going to be held in the water in your body. And it gets rapidly distributed through the blood. To remain constant, the amount of heat that's lost should be equal to the amount of heat that's generated. So to lose heat will be exchanged with the environment. And there are several different ways in which heat is exchanged. First, there's radiation. Radiation is when objects warmer than the environment lose heat. So when your body is warmer than the environment, the heat from your body will radiate off from your body into the environment. More than 50% of your heat is lost this way. The second type is convection. Convection is heat loss as air moves across your body. As this happens, the air warms and carries it away, and then your body becomes cooler. About 15% of your body's heat loss is through this method. The third method is evaporation. Evaporation occurs when water changes from liquid to a vapor. There's two types of what we call perspiration. There's insensible perspiration. And this is water loss across the epithelia, or in other words, across your skin. This is a constant type of perspiration. About 20 to 25 mils per hour are lost this way. And then we have sensible perspiration. Sensible perspiration will lose about 0 to 2 to 4 liters per hour. This is highly variable. That's what I mean by 0 liters per hour or 2 to 4 liters per hour, right? So an example of sensible perspiration is sweat. Of course, sweat is going to increase when you're more active or when it's really hot out, and then you'll get towards that higher range. And if you're not doing any activity and not too hot, then you're going to be losing about 0 liters per hour of fluid that way. The last type of heat exchange with the environment is called conduction. So conduction is a direct transfer of heat between objects. So if you, for example, if you sit on a cold chair, right, the chair is going to become warm and your butt's going to become cold. The heat transfers from you to the chair. There are heat loss and heat gain centers in your hypothalamus. And the heat loss center is activated when your temperature rises. So the temperature has to rise above a set point. You have a set point. 98.6 is your normal body temperature. There's a little bit of a range in there. And if your body temperature rises up above that set point, then the heat loss center is going to become activated. This will cause peripheral vasodilation, so more blood will move to your skin. It sends that warm blood to the surface. Your sweat glands get activated so they can take the fluid from the blood vessels and produce sweat and secrete that out through the ducts of the sweat glands. And your respiratory centers become activated. So you're going to increase your rate and depth of breathing. So by breathing out that moist air, you're breathing out that water vapors, and that also will increase evaporation through the lungs. Also in the hypothalamus, you have the heat gain center. The heat gain center prevents hypothermia. It's activated when the temperature is below acceptable levels. So it gets activated with decreased temperatures. If your temperature drops below the set point. In this case, the sympathetic nervous system gets activated. The sympathetic nervous system will cause vasoconstriction to your skin, so you won't lose that heat through the sweat glands. Instead, blood gets diverted to your deep veins. And it traps your heat in the core of your body. When the heat gain center is stimulated, it will stimulate a process called Shivering Thermogenesis. Shivering thermogenesis is when you are shivering to produce heat. Shivering increases oxygen and energy consumption. So you have to use energy. You need oxygen and you need energy in order for those skeletal muscles to shiver. And so since you are creating more energy, you're creating more ATP, that means you're also going to create more heat. Heat through this method will warm the deep blood vessels and it will increase heat generation by about 400%. So it's a pretty good method. Shivering thermogenesis does this by stimulating the sympathetic nervous system. And then the sympathetic nervous system stimulates the adrenal glands. And then the adrenal glands release epinephrine. And then epinephrine stimulates glycogenolysis. If you remember what that is, that's breaking down glycogen into glucose. And then glucose can be used to produce ATP, which also produces heat. Another type of thermogenesis is called non-shivering thermogenesis. So non-shivering thermogenesis is heat that's produced without shivering. It will increase your heat production by 10 to 15%. But it takes weeks. It's not something that happens immediately like shivering does. This involves hormones. It involves the release of hormones. So, for example, the thyroid releasing hormone gets secreted from the hypothalamus. That causes the thyroid stimulating hormone to be released from the anterior pituitary, and that causes thyroxine to be released from the thyroid gland. And when thyroxine is released from the thyroid gland, that will increase the catabolism in all the cells in your body and the catabolism of nutrients like glucose. So then you're generating ATP. And if you're generating ATP, that means you're also generating heat. There is individual variation of heat production. What I mean by that is that, first of all, people can acclimate to their environment. Take, for example, people that live in a place like Tierra del Fuego. Tierra del Fuego is off the southernmost tip of South America, across from the Strait of Magellan, and it's a very, very cold climate. So in this place, residents live naked in the snow. So they have acclimated to this very cold environment, and it really doesn't bother them that much. But then you look at people that live in a place like Hawaii, or even a place like Peru, and in the winter, when it gets to be about 60 degrees or so, they will wear sweaters because they feel it's cold. So they've acclimated to the warmer environment. Another example of acclimatization is body size. Small individuals, they're going to lose heat much more rapidly than larger individuals. So now I just want to discuss two different types of fat that you have in your body. There's brown fat and there's white fat. We'll look at brown fat first. Brown fat is rich in heme, right? And heme is part of red blood cells. And it has cytochromes in it and mitochondria. So the heme gives it sort of a reddish, a deep, rich color. And it has an increased ability or an increased capacity to generate heat. So instead of the cells capturing the heat like we see, so glucose releases ATP when it catabolizes and heat. So instead of this occurring, the cells are going to release it to the surrounding tissues. So this is what happens with brown heat. So glucose produces ATP, but then the heat that it releases is not captured by the cell, it's released into the surrounding tissues. From there, that heat will move into the bloodstream and get distributed throughout the body. Newborns have mostly brown fat, and so the heat that's generated by their cells gets released into their tissues and it keeps their body warm. But then it's cooled down quite quickly through those methods that I showed you, how heat is distributed from the body to the environment. And so then the babies lose heat very, very quickly. So they have to be dried, kept dry and bundled. The brown fat for newborns, though, speeds up their heat generation by 100%. Adults have brown fat mixed in among their white fat. This happens because with age, there is an increase in body size, and we have more skeletal mass, and increased skeletal muscle mass. And so with all of these things, then shivering thermogenesis becomes more effective. White fat is the fat that most of us are more familiar with. This is the type of fat that stores energy, stores triglyceride in these big oily droplets throughout the body, and in large quantities can cause obesity. In healthy, non-overweight individuals, adults, white adipose tissue or white fat composes as much as 20% of body weight in men and 25% of body weight in women. White fat does generate heat, but it generates heat at a rate of 1/3 of other tissue. Which means it's not being catabolized very quickly, which makes it a very good insulator. As far as body temperature goes, individuals have different set points in the hypothalamus. For most people, temperatures are going to peak during the day and the early evening. And then at night, they fall about 1 to 2 degrees Celsius. This, for women, it fluctuates during the ovarian cycle. And we'll see when we get to the reproductive system that at ovulation, the temperature will drop 1 to 2 degrees Fahrenheit. I just want to mention heat exhaustion and heat stroke. So heat exhaustion is a condition when a person has been exposed to high temperatures, particularly when that's combined with high humidity. As well as strenuous physical activity. With heat exhaustion, there's excess fluid loss through sweating. And this occurs because the high temperatures stimulate the sweat glands to release sweat so that the temperature drops. But this also means that blood pressure is also going to drop because fluid is being lost. In heat exhaustion, the blood moves to the skin because the blood vessels are vasodilating, which means blood is moving away from the brain. When blood moves away from the brain, it can cause things like nausea and headaches, and a person might collapse. A person experiencing heat exhaustion needs fluids, salts, and they need to cool down. They need to be moved into a cool environment. So with heat exhaustion, the heat loss center was stimulated. If heat exhaustion becomes severe enough, a person might go into a heat stroke condition. And this is where the thermoregulatory center stops working. And so then the heat loss center isn't working, and so temperatures rise uncontrollably. The skin starts to get hot and dry. Whereas with heat exhaustion, the skin would be sweaty and cool. The body temperature might rise to 106 to 113 degrees Fahrenheit. At these temperatures, cells die in the brain, the liver, the skeletal muscles, and the kidney. There are a couple of conditions that will predispose a person to heat stroke. diseases like heart disease or diabetes. These conditions both affect peripheral circulation. Without good peripheral circulation, the body will not be able to release the heat, and the heat will build up in the body and lead to heat stroke. A fever is a body temperature over 99 degrees Fahrenheit. A transient fever is a fever that occurs with exercise. So we see this a lot in young children who are out playing in the playground and just working up some heat. And we can also see it normally in adults who have been exercising excessively, like after a marathon. These things will increase the body temperature up to 103 to even 109 degrees Fahrenheit. Those types of transient fever will go away as soon as the exercise stops. But another type of fever can occur with pyrogens. When your body cells are fighting off an infection, your T cells will release interleukin-1. That's a cytokine, and that interleukin-1 is a pyrogen. Like I mentioned earlier, fevers can occur from heat exhaustion or heat stroke. Fevers can also occur from drug reactions. Any drug reactions that impair the sweat glands. An example of these would be an anti-muscarinic drug. If you remember, the sweat glands have muscarinic receptors on them. So if you take an anti-muscarinic drug, that drug will inhibit the muscarinic receptors, and therefore you can't sweat. And if you can't sweat, you're not going to be able to release heat, so your body temperature will increase. Another thing that could cause a fever is congestive heart failure. Congestive heart failure will restrict your blood flow. If you restrict your blood flow, then blood won't move to the skin, and you won't be able to release heat through sweat. So heat exhaustion and heat stroke are conditions where the body temperature gets too high, but there is a condition when the body temperatures get abnormally low, and that is called hypothermia. Hypothermia is below normal body temperature. In hypothermia, the thermoregulatory centers become less sensitive. So the heat gain center isn't working. If the heat gain center isn't working, and as the body temperature is dropping, then the cardiac output decreases because the heart rate decreases and stroke volume decreases. The heart doesn't have to push blood to the tissues because the tissues aren't active, so they don't really need additional oxygen or glucose. And also the respiratory rate decreases. Again, the cells don't need oxygen if there's no activity there. If the body temperature gets below 82 degrees Fahrenheit, then cardiac arrest will occur. There's no heart rate, there's no heartbeat, there's no respiratory rate, there's no response to painful stimuli. The person may be assumed dead. But in actuality, a person that has gone into hypothermia can still be resuscitated hours later. A person like this would need gradual warming. Children who drown in cold water can be revived up to 4 hours later. However, a person who is frozen can't be revived. The reason for that is that water is going to expand when it's frozen, and when the water in the cytosol expands, it will destroy the plasma membranes. Okay, this ends our lecture on metabolism, day 2.